Okay, so today we're going to look at brain development, and particularly focusing on language acquisition, so how that happens. We'll come on to the mechanism of that, which is called neuroplasticity, so the fact that basically your brain is changing and evolving depending on what senses the sensory information that you receive. We'll also get on to look at how genes affect development and how this can affect behaviour, and the problems that we have with particularly stress and how that as a child can put you at increased risk of mental illness later on in life. Okay, so first off, what is neuroplasticity? This is just how the brain can change and adapt to new situations. So basically your brain is capable of learning. So it receives sensory information, but of course we can have damage which can cause problems, so something like a stroke, which you've looked at before. Okay, so first off, neuroplasticity, so it occurs throughout life. It's probably faster when you are younger, but it leads to obviously a healthy brain. So first off, what synaptic plasticity is. So if you remember the structure of a neurons, oops, that's not a very good one. So we have the cell body, we have the myelin sheath to insulate it, and of course we have the axon here. And they have lots and lots of different connections called dendrites and nerve endings. So if we have another nerve cell here, like so, and these are connected up, like so, then what happens is that the more that a pathway is used, so if lots and lots of nerve impulses travel along this pathway, then we will have what are called bifurcations. So bifurcation just means splitting in two, and these will form more synapses. So we'll be able to take heavier and heavier traffic, and that's what we mean by synaptic plasticity. However, if this pathway is not used very much, then what happens is we have something called synaptic pruning, and this is when you would lose the synapses between them. So as you can see, it's the more it's used, the more synapses are formed, and the less it's used, the less synapses are formed. So as we can see here, you start off with about 2,500 synapses. By the time 2 or 3 years old, up to 15,000, but then that is gradually pruned back. So the ones that are not reinforced weaken, and we lose those connections. Okay, so what do we have here? So these are fine-tuned throughout life, but the key thing here is that early childhood is the critical part of development. And if the nervous system doesn't get the information that it needs at this point, then it can never fully develop. Okay, right. Okay, so adolescence, which is what you're in at the moment, so you have lots and lots of pruning back going on within adolescence, so only your stronger connections will be used there. So non-synaptic plasticity, this refers to basically changes in the number of ion channels that are on the synapse, which receive the information and send it on to the next neuron. Right, moving on to the development of the language area of the brains, we have these things called phonemes, and these are the sounds produced by people speaking. So the, all of those are phonemes, and you're able to respond to these within the womb, and so you actually begin to learn language before you're born. There's this wonderful fact here, there's a thing called parentese. For instance, ooh, isn't he sweet? Ooh, all of that you use for very young children. And incredibly, this has even been observed in rats. So rats go sort of goochie goochie goo to their offspring as well. So as you hear these phonemes, you have different neurons responding to the different sounds. And if you don't hear these critical sounds at an early age, then you never form these necessary synapses. So this is why it's very difficult to distinguish phonemes in other languages, those that don't exist in English for us. So The development of language requires lots and lots of experience to be able to decode sound and work it out into what it means in language here. And again, this has to occur in early life. Most babies start producing sounds at a fairly early age. Obviously, deaf ones tend to be a lot behind with this. And incredibly, by the age of 2, you've basically learnt, or your capacity for learning phonemes is finished by then. And after that, then you learn the rules of grammar in terms of verbs, etc., things like that. After this time, you can pick up new vocabulary. Obviously, you do that throughout your life, but it is impossible to learn those rules of language. And we know this because there are, unfortunately, children that have had a very, very poor upbringing early on in life. We've got an example here of a girl called Jeannie, and she basically wasn't spoken to at all until she was 13. When the interventions were put in place, she had great amount of training, but because she wasn't exposed to a critical time when her brain was developing, then she never got past a critical a basic level of being able to communicate. Roughly about 7,000 genes are expressed in your brain, so that's about a third of the 20-odd thousand that we have in there. And what we can have to these is these things called epigenetic modifications. So quite simply, this can be if you have sort of a length of DNA, A, C, G, T, C, G, A, T, for etc. I've got put T at the beginning, so I've got groups of 3. And what can happen is just a methyl group can be tagged onto these bases, and that basically prevents the DNA polymerase, the enzyme, moving along. It would be blocked by that, so the gene couldn't be expressed. We also have these things called histone proteins, and your DNA, we used to seeing it as the classic double helix like that, but in reality, the DNA is actually wrapped up around these circular proteins called histones, and round and round like that, and then you have areas where the DNA is not wrapped up. But where the DNA is wrapped up around these histone proteins, then it is much more difficult to express. So again, you can switch off the genes through methylation or by this histone, binding of histones there. So what influences your language? Well, we have things like famine, exposure to drugs when you're in the womb. There's something called fetal alcohol syndrome, which severely affects brain development and affects your behavior throughout life. Examples of residence of stress-related genes in the hippocampus receive good parental care. And it is even possible that things that happen in your lifetime can get passed on to the next generation. Okay, in psychology, we have a real debate over the relative influences of genes in the environment. And the best way of studying these is to look at what were called twin studies. So in a twin study, you have 2 types of twins. You can have what are called monozygotic twins, whereby one egg is fertilized by one sperm, and it then splits into 2, which then develop into 2 identical twins. Or we can have digozotic twins, in which you have 2 different eggs and 2 different sperm. So what we have here, these are as related as brothers and sisters, whereas these are obviously genetically identical. So we're seeing something like schizophrenia, then monozygotic twins, we have a concordance rate. So concordance means if one twin has it, then there's a 40% chance that the other twin will have it for schizophrenia, whereas in dizygotic, we only see 7%. So this shows that there is a strong genetic influence in the 40%, but obviously if it was totally genetic, then it would be 100%. So we've got a big environmental influence as well. And what most psychologists think about is we have something called diathesis stress, which is that you inherit a disposition for mental illness, but it may or may not be triggered depending on the environment that you live in. If we study abused children, then they are 50% more likely to suffer from mental illness and things like depression than adults, and they also find it harder to recover as well. Adults that are subjected to childhood abuse are also at greater risk of other things like schizophrenia, eating disorders, etc., etc. And of course, victims of abuse are much more likely to end up abusing themselves and abusing things like drugs and alcohol. And one of the key theories is that if you experience trauma during early times, during your early development, then this could turn some of the epigenetic switches we were talking about earlier, which alters your gene expression. One hormone that we know is very much related in stress is this hormone called cortisol. So if we look at the sort of structure of the brain here, so the brain, the spinal cord, we have the hippocampus here, and this releases, sends impulses to the hypothalamus, like so. The hypothalamus then releases two hormones, which cause the pituitary gland to produce adrenocorticotrophin into the bloodstream. This then travels to the adrenal glands, which sit on top of the kidneys, and cause the adrenal glands to release cortisol. Cortisol then goes back into the bloodstream, and it binds to these receptors in the hippocampus up here. So the cortisol goes up to there, and this then stops it from releasing the corticotrophin releasing hormone. So what we have here is a negative feedback mechanism, so it will in effect switch itself off. And what we see is that this prevents the excessive release of cortisol. But what we do see in the people that have received real sort of childhood trauma, then they don't really have this feedback loop. So as a result of this, without the negative switching off of it, we end up with more and more of the stress hormone being released from the adrenal glands and causing all the problems with that, increasing stress, etc. Okay, so what you should be able to do at the end of this is to describe briefly plasticity, neuroplasticity, language development in insects, explain the roles of genes in the environment, use the evidence from, as I said, the mono and dizygotic twins, the diagram like I've just produced here to show negative feedback and how childhood abuse could affect cortisol levels.